Good morning, everybody. You are the VIPs, you are the ones that come the first session of the last day, so thank you for that. That's extremely important for us because I think we have a very good program for this last day, and basically this morning we have some kind of a summary about what the ETPN tries to do. So we'll have excellent two keynote speakers. The first one indeed is Professor María José Alonso from University of Santiago de Compostela. She will talk indeed about, let's say, drug delivery for oncological drugs, and then after Olivier Guénat will be on stage. Then we will have, let's say, two very interesting talks that I think most of you are waiting for about the preparation of Horizon Europe by Heiko Frima from the European Commission, and also from the NCP Spain for the let's say the last calls of the current H2020 projects. And then we will have what was supposed to be a brokerage, but we have received many, let's say, few proposals. I think it's because of the success of the direct face-to-face meetings you had through the B2 match. So thank you for having using a lot. It was the first attempt, and I think that we will continue this because it has worked quite nicely. But first, let's start by the first keynote of this morning. So Professor María José Alonso, if you come on stage. Thank you very much for being here.
Thank you, presentation. Good morning to everyone. Thank you for being here today. I mean, I was feeling a bit bad yesterday when everybody was having fun with that wonderful dinner, and I said, where is the bus? We have to leave at 10:30. I really wanted you to be here, and I wanted to get some rest before giving my presentation today. So anyway, thank you for being here, and as Alexander said, I am based at the University of Santiago de Compostela, only 2 hours away from here. And today I would like to talk to you about multifunctional polymeric nanocapsules, targeted delivery oncological drugs. And many of you may say I was just talking to Lorena. By the way, thank you very much for inviting me, Lorena. And also thank you to the ETPN. And I also want to say that I am delighted to see that we have such an outstanding team, the new members of the new chairs of the ETPN. I think this is amazing that so wonderful people were interested in being part of this platform. So anyway, going back to the subject, I was just yesterday wondering, oh, wow, everybody is working on cancer. Everybody's trying to deliver oncological drugs. Are we getting there? Because, you know, more and more people talk about whether nanomedicine has delivered according to the expectations, and many people are very critical about nanomedicine, and in particular in the oncological field. Well, if we look at the literature, we realize that, in fact, even though nanomedicine started in the 60s, according to my view, it has only been on the last decades that people have really dedicated significant efforts, that we had a massive community working in the field. And we still know little, according to my view, about, especially about how nanomaterials interact with the biological system. So I do believe that we need to work more intensively, more coordinated, more in collaboration with biologists, and to keep in mind that nanotechnology is there mainly to solve problems. So I always say the main mission of my lab is to solve problems of drugs, problems of treatments. We want to make better treatments. And my background is pharmaceutical technology. So the expertise in my lab is how to link, how to put drugs into nanocarriers. And one of the fields is cancer, because we do believe that cancer is already benefiting, but should benefit much more from nanotechnology, from nanomedicine. So this is my research group. We are around 25 people working in this research institute on molecular medicine. We are in the 2 upper floors, and we are also associated to another research institute. It is linked to the hospital. Of course, I'm linked to the School of Pharmacy, where I teach biopharmaceutics and radiopharmacy. So thanks to all of them because they are the main actors, the main responsible for the work I'm going to present today. So I normally like starting my presentation with this slide, with these photos, where we see different kinds of nano particles, nanocapsules. And mainly what I want to highlight here is that these nanocapsules are composed of a core surrounded by a corona, by a shell. So I said before, our aim is to solve problems, and we try to design rationally these nanocarriers in order to solve problems of specific drugs. So the way we design these different nanocarriers is taking into account the nature of the drug that we want to load into the nanocarrier and the biological barriers that we want to overcome. Okay, so we have to define the structure, we have to define the composition, taking into account mainly these parameters. So the materials that we are using are mainly materials that are recognized for their safety profile. So we normally try to avoid new materials which have not been proven for the safety because of our translational vocation. We have to say that we are quite limited in that regard, and sometimes we explore new materials as well. But basically we use polysaccharides, polypeptides, oils, surfactants, very classical excipients. So the key here is to be able to find the right combinations and combine them in the right way in order to achieve our goal. So we say we use nanotechnology in order to help drugs overcoming biological barriers. So what kind of barriers are we talking about? We are talking about not only cell barriers. I often say that, in my view, people working on nanomedicine has put too much emphasis on the interaction of nanoparticles with cells, and we have not considered sufficiently the physiology, the anatomy, and the fact that before reaching the cell, the drug, the carrier, has to confront very significant barriers, and the cell is just the last one. So we have been working on overcoming systemic barriers such as the biodistribution, the interaction with proteins. But we have also dealt with other, even more complex barriers, trying to facilitate the transport of drugs across, for example, the ocular mucosa, the nasal mucosa, the intestinal mucosa, and also across the skin. So using nanotechnology, you can help drugs overcoming all these different kinds of barriers. So regarding the therapeutic areas in which we are now currently involved in my lab, I would like to highlight the possibility to deliver orally peptide molecules, and this is the result of our participation in the Transient European Consortium that I coordinated, and we looked at how nanomaterials interact with the GI tract. We have dedicated significant efforts to the area of vaccination, and nowadays we are working with different vaccines, either for therapeutic purposes such as multiple sclerosis or for HIV. We are actually quite advanced working with HIV, and in fact, Iraida is around. She has coordinated this nanopilot European project, and we have been able to scale up our prototype, which is an HIV vaccine candidate. And we have also worked in collaboration with the NIH in the US and Canada and investigated the behavior of this nano vaccine against HIV in macaques. We are now aiming at going into clinical trials. We are also trying to deliver drugs, not regular drugs, but mainly polynucleotides, RNA across the eye. And other areas include dermal drug delivery. Here, we just applied for a FET Open project. Let's see, we should have the results soon. So we are trying to combine laser application with nanotechnology. We are also working in trying to design new meniscus in tissue engineering, and the name of the European project is Mephisto. We are also working in the Be Smart project with Raymond Schiffler, who was around yesterday. And in this case, what we are trying to do is to deliver RNA to the brain, nose to brain delivery of RNA, And finally, we are working in the area of cancer. So I was doubting what should be the focus of my talk today among all these therapeutic areas. But because we have particularly benefited from the ETPN in the area of cancer, we have participated in 3 different Euronanomed projects. I thought it would be nice to tell you the story of what we have learned over the last decade working in these Euronanomed projects. So this is a very easy illustration of the nanocarriers that I showed you before. So just for you to know what kind of technologies we are using for producing these carriers, we are mainly playing with hydrophobic interactions and ionic interactions. We are helping materials to self-assemble. And so we have these kind of structures in which we have a core which can be an oily core or a solid core, and the core is surrounded by a number of layers. It may be only one layer, it may be multiple layers, depending on the final purpose of the formulation. In some cases, we also want to functionalize the surface layer in order to target the carrier, which is what I'm going to show you today. So there are a number of interesting key features in the sense that the technology is very versatile, solid liquid core, multiple layers. Obviously, we can modulate the surface charge of the system, and we can modulate the size. It's a kind of toolbox flexibility in the sense that we can incorporate one or more active ingredients, protective agents, controlling agents, targeting agents. So let's go onto the topic of today, focus on cancer. What is the problem to solve? I don't have to tell you what is the problem for the treatment of cancer, right? And I am sure that you know that even though in the last decade there has been a lot of emphasis on the new targeted therapies and immunotherapies, there are still very important problems that remain to be solved. If we think about the classical chemotherapy, you all know that the efficacy is low, the toxicity is high. We cannot reach the metastatic cells. María de la Fuente gave a nice talk yesterday in that specific subject, trying to reach metastatic cells, the resistance problem. But then thinking about monoclonal antibodies, because this is the fashion thing nowadays, using monoclonals for treating cancer. And some people think that we have solved the problem of cancer. This is not at all the case, not with immunotherapy either. So just for you to know, we have at least 35 marketed monoclonal antibodies, and more than 2,000 in clinical trials. And what is common for all these antibodies? All of them are intended to target extracellular targets. None of them goes to intracellular targets. So we are missing a big space there for the treatment of cancer. And in addition to this problem, we cannot discard the important immunological reactions that we are seeing now, and already the resistance phenomena as well with these kind of therapies. So we are improving, but many problems to solve. So I told you before that I want to share with you what we have learned in 3 European Euronanomed projects, LymphoTarg. We design systems to target drugs to the lymphatics. Niche, we were trying to deal with the immunogenic cell death concept, and 2 intra-target that is going on nowadays, trying to reach tumor-associated macrophages, as Fernando Torres presented yesterday, but also cancer cells. We have also got financial support, significant financial support from the Spanish government and from the World Cancer Research Organization, UK. I have to say that the results of this project have been presented to the NanoMed tab, and we were selected, and actually that was the origin of a nice story. Let's see how it ends. But at that time, it was already like 4 or 5 years ago. We were encouraged to start up a company based on the data we had at that point. We have continued accumulating data, and we are almost ready to start the company, but not yet. I'm still quite reluctant about making this step ahead. It's not an easy thing, but anyway, participating through these Euronanomed projects, and then with with the NanoMed tab, we have been going into, I believe, the right direction. Let's see if we get there. So our delivery carriers, we call them multifunctional polymeric nanocapsules. Well, polymeric nanocapsules, it is obvious why we call them that way. Multifunctional is because they have different functionalities. So the conceptual mechanism means that we have a dual targeting, tissue targeting, and dual cell targeting. When we talk about targeting, very often people talk about cell targeting, and we forget about the tissue. And as I said before reaching the cell, the carrier needs to reach the tissue, and that is the main problem, in my opinion. So we say dual tissue targeting. What are the tissues? Of course, the tumor, and for us, the tissue is the lymphatics as well, because in many cancers, metastatic cells are accumulated in the lymphatics. So we want our particles to go to the tumor and to the lymphatics. And then once they reach that level, we want them to go to cancer cells and also to tumor-associated macrophages. So multifunctionality. So some key structural parameters of our nanocarrier that is illustrated here. We have what we call a biofunctional polymer around the system. We have an active targeting ligand, and we have a special nanoconstruct architecture. The system is flexible, is elastic, and that is also an important property. Size can be adapted, can be tuned. The materials, as I said before, are regulatory acceptable materials. The technique is highly reproducible, and we have loaded a great variety of drugs within this nanocarrier. Easy to scale up. We have also done some medium scale up, and it can be freeze-dried. So basically, all the pharmaceutical properties that we need for such a delivery carrier. As said, we have already encapsulated a variety of drugs, small molecules such as docetaxel, paclitaxel, aplidin, and now I'm going to show you more the details. So this is more in detail the system. For example, in the case of docetaxel, it was dissolved in this oily core, and we surrounded this oily core by these hydrophilic polymers, polysialic, hyaluronic acid. We functionalized the polymer with peptide targeting ligands, for example, TLIP1, and because this targeting ligand has specific receptors in tumor cells and also in endothelial vessels, and it also helps the tumor penetration, which is also one of the big barriers for the nanocarriers to reach the tissue and to cross the stroma before reaching the cancer cells. So we have been taking into account all these barriers. So here we have the percentage of the drug per gram of tissue. This is docetaxel in the tumor following administration of 3 formulations. Taxotere, nanodocetaxel, we say classical. These were the results of the first Euronanomed project, so hyaluronic acid nanocapsules. And these are the targeted ones, MPN. So you see that with our targeted nanocapsules, the amount of drug that reaches the tumor is amazing, is very, very high. Actually, we never saw such a high concentration in the literature. Look at this. When we administered Taxotere, we reach around 2% in 1 hour, but at 24 hours, we have almost nothing in the tumor. And the same happens in humans, in fact, as far as we know from the literature. Only a very minor amount of the drug goes to the tumor. When we administer docetaxel in our nanocarriers, we increase 37 times the concentration of drug in the tumor, and this is a dramatic accumulation. So the same is illustrated here. If we compare the accumulation in the tumor with the accumulation in other relevant organs, we see a dramatic difference. Clear targeting. The drug is targeted to the tumor, to the lung tumor, I have to say metastatic lung cancer. And it also accumulates significantly in the lymphatics, where the metastatic cells are located. So here we have the pharmacokinetics, where you can see that normally when you have a passive targeted delivery carrier, you increase the half-life of the drug, right? So here we are not talking about increasing the half-life of the carrier. The half-life is, well, it has increased a bit, but not much. What we have increased significantly is the volume of distribution and the MRT, which is in agreement with the targeted approach that we are claiming here. So no passive targeting is mainly active targeting. And some people told us, oh, yes, but everything accumulates in the lung because this is the first time where everything goes. Maybe your particles are aggregated and they remain there. So we did the same kind of experiment in healthy mice, and we could see that the accumulation of our MPN technology was very low in regular mice. So it only accumulates if there is a tumor in the lung. So the response, as usual in terms of tumor growth, we can see that with our formulation, the tumor growth is highly reduced over the time. There is almost no tumor growth. But more important than that is, this is in metastatic lung cancer model, orthotopic, and this is in a pancreatic cancer model. In this case, the person with whom we were collaborating, Dr. Manuel Hidalgo, he wanted to confront our formulation with Abraxane because he participated in the clinical trials of Abraxane and he really likes Abraxane, and he said, we want to confront your formulation with Abraxane. So we administered Abraxane, and you see at this point the animals had to be sacrificed because their general status was really bad. However, when we administered our formulation, the tumor size went down and the animals looked in a much better shape than following administration of Abraxane. I remind you, Abraxane are nanoparticles made of albumin containing paclitaxel. And then what we wanted was also to reach metastatic cells. And this is what you see here. When animals were not treated, they had to be sacrificed at 37 days. Those treated with Taxotere, the commercial formulation, survived up to 47 days, but in very bad shape. And finally, those treated with our nanocarriers could survive in a very good shape with almost no metastases. So clear reduction in the metastatic load and also clear reduction in the tumor size. So this is about small molecules, targeting small molecules, trying to increase the efficacy of the classical chemotherapy and trying to reduce the toxicity, okay? I didn't have the time to present the toxicity data and everything, but we have done tons of experiments with different animal models. So then the other idea, a new concept, a paradigm shift. As I told you before, all marketed monoclonal antibodies are intended to target extracellular targets located on the membrane or located in the stroma, like it is the case of immunotherapy, okay? Why nobody's trying to reach intracellular targets with monoclonal antibodies? Simply because they cannot cross the membrane. So we wanted to explore the potential of nanotechnology for delivering monoclonal antibodies inside the cells. And this is a new concept because up until now, monoclonals have been used as targeting ligands, but to target receptors on the membrane, but never inside the cell. So that is new, and this is what we wanted to prove. And for that, we chose a specific target, gasdermin, because gasdermin B, because the possibility of a collaboration with Gemma Moreno from the University of Madrid. And she is a great expert in this target, and she said, well, I wish I could have a monoclonal antibody going in towards this intracellular target because they have a lot of information about the target. They know that overexpression of gasdermin is involved in cell migration, tumorigenesis, and it is related to the resistance in anti-HER2 drugs, the classical targeted therapies and protease kinase inhibitor therapies. So she really wanted to deliver a monoclonal antibody against this target. So we collaborated with them, and we first investigated the internalization of the antibody. And this is what you see here. These are lung cancer cell line cells, A549 cells. And you can see some little spots, green spots, which correspond to the monoclonal antibody. So we have the same kind of images. Actually, this work was just published. I mean, I think it's not available yet, but it will be in the next few days in Clinical Cancer Research, where we see the monoclonal antibodies inside the cancer cells. And we found the same behavior in a variety of cancer cell lines. So it was clear that the antibody could go inside. So then the next question is, what is the mechanism of internalization? So, well, as expected, the mechanism was an endocytic pathway which occurred at 37 degrees. It was stopped at 4 degrees, and it went back again at 37 degrees. You can see here how at 37 degrees, 80% of these cells were positive, had antibodies inside. At 4 degrees, the process did not occur, and when we moved from 4 to 37, the process started again. So an endocytic pathway mediated by the temperature. Once we knew about this endocytic pathway, we were wondering, so what is next? What happens inside the cell? So we know that when there is this endocytic pathway, very often the system ends up into the endosomes, and then the drug is degraded. So we wanted to avoid this intracellular degradation, and we looked at the endosomal pathway, the endosomal escape. So we labeled the antibody in green, we labeled the endosomes in red, and we could see that after a few hours, the monoclonal antibody was escaping from the endosomal compartment, and we were happy with that. Very important, I told you before, it's not only the cell, it's the tissue. We have to make sure that our nanoparticles are stable in the bloodstream and that they are able to reach the tissue. So working with cells is easy, but then what happens in vivo? So we performed this biodistribution study in this orthotopic breast carcinoma model, and this is a cross-section of the tumor. And again, we could see the monoclonal antibodies inside the cancer cells of the tumor. As I said before, before going into in vivo, we checked the stability of our nanoparticles in plasma, and we did find that they were quite stable for up to 24 hours. And finally, we administered these particles in vivo in order to see the response towards this antibody. And this is the experimental protocol. We administered 2 doses a week for 3 weeks. The dose was 17 milligrams per ml, a pretty low dose, in fact. And we found this data. Here we are representing the tumor growth over the time. These are 3 control formulations. Basically, we were not expecting any response, and this is the response observed for these nanocapsules loaded with monoclonal antibody. We are now trying to improve this profile because, as I said before, from our knowledge, this is the first attempt to deliver monoclonal antibodies inside the cells, showing in vivo data. There are a few papers in the literature showing only in vitro internalization, but no in vivo data. There are other strategies that biotechnology experts are investigating, such as the design of nanobodies or single domain antibodies, things like that in bioengineering. But using a very simple technology as the one presented here today, we believe we are the first reporting such a formulation, relatively easy formulation approach. So just, I think I have spent more time than needed probably, but some key messages is like, when we talk about active multi-targeting technology, we have to take into account that we have to target the tissue before targeting the cells. So first the tissue. We should not assume that because we are targeting cells, reaching the tissue, that sometimes people say, okay, you put a targeting ligand on the particles, this targeting ligand goes to the cells, so the particle knows where to go, and it will go there. No. Before it has to reach the tissue, and then once it is in the tissue, the cell targeting may work. Okay, so we have validated in vivo for the first time the intracellular delivery of monoclonal antibodies. We have significant proof of concept studies. We have the possibility, we have done already, to combine small molecules, either cytotoxic drugs or protein kinase inhibitors in combination with monoclonals. We know that the technology is scalable, and we know that these particles can be freeze-dried. And just before finishing, well, again, to my research team, by the way, all the work done with docetaxel was done by a bunch of people, but also in collaboration with Noemi Chabak, who is around, and all the work done with the monoclonal antibodies was done in my lab, mainly by Ana Cadete and Desiree Tejero and her team. So just before finishing, if you have not registered yet to the annual meeting of the Control Release Society, which is going to be in July, you are still in time. I think early bird registration ends this week. So please join us there. If you don't know about the Control Release Society, visit our webpage. I am the president, so I have to do some publicity, but it is going to be an amazing conference. We have already 1,000 people registered. But for those who cannot make it in July, because that meeting is 21, 23 July, I am already announcing the 25th anniversary of the Spanish-Portuguese local chapter of the Control Release Society. And we are going to have our meeting in January 22, 24. Bruno Sarmento, my colleague Dolores Torres, and others are involved in the organization of this event. And we hope to see many of you there. Another announcement is about this journal, Drug Delivery and Translational Research. I am the editor-in-chief. It's an official journal of the Control Release Society. So I invite you to submit your materials, especially if you are interested in translational research, to the journal. Thank you very much for your attention.
Thanks a lot, Professor. That was amazing. So I'm sure we will have many questions. Who will be the first one? Raise your hand.
María, very nice work. Congratulations. And one of my questions is that, because you talk about the first target tissue and then the cell, but when we look at the taxol you used in the health mice, actually the targeted particles didn't go to the tissue. It's the non-targeted particles that go to the tissue more than the targeted nanoparticle.
No, actually, we can show the slide again, but the standard deviation was very big. It was the same.
So it's not significant.
Actually, none of them went because the accumulation was very low.
Okay, and the other question is that I saw that you have more than 37-fold increase for the tumor uptake than the clinic drug. Did you check the difference between targeted nanoparticle and the non-targeted nanoparticle?
Yes, actually, we can go over the slide, but for the sake of time, for the targeted nanoparticles, we had reached 20% of the drug was in the tumor in 24 hours. In the same time, only 4-5% of the drug was for the non-targeted particles, and 0.2 was for the commercial formulation.
Okay.
So it was a very high difference.
So the tumor specific is not only the targeting group, it's also the formulation or the material.
It's everything together, yes.
Mm-hmm. And finally, I saw that you have, after the time, you have the drug resistance. Because after 30 days, and the tumor grows back, come back. For the monoclonal, you mean? No, for the taxol. Well, not really. No, we can go back to the slides. There is not a subsequent increase. Let me go first. In this case, you mean? Well, in this case, we only gave a couple of doses. Then we did another experiment in which we gave more doses of docetaxel, and it was okay. So we cannot consider this a phenomenon of resistance at this point, according to the expert. I'm not expert on resistance. And then in the other case, yes, we didn't eliminate
completely, but almost completely, the growth. Yes?
PDX models they grow very slowly.
Yes.
Yes, so because they go very slowly, so this curve means some part the drug resistance. Because usually the PDX model, if you take the aggressive model, they will grow very fast.
Well, this is a PDX model, in fact, yes. But I cannot discuss about resistance, honestly. According to the expert Manuel Hidalgo, that was not the case. There is no resistance here, but he said that we were discussing about that. And he said that we will have to stop this treatment, start again, and see if there was no response in order to prove that. Mm-hmm.
Other question? Okay, because if not, I have one.
Thank you. I agree with you with regards to the intracellular targets because it opens, a wealth of additional possibilities. But having to evaluate the efficiency of targeting something that is outside and something for which, on the contrary, you need to overcome certain barriers. Give me a number. So how big is the loss of efficiency in the targeting of an intracellular target in comparison to the targeting of
an extracellular target?
Yeah.
Well, obviously, reaching the intracellular targets is an additional barrier. At the end of the day, what we want is to have efficacy reaching this target. I don't think we can provide a number of how many molecules can go outside or inside. If you could see that once the particles reach the tumor, most of them will go into the cell. So at the end of the day, what you want is to have efficacy and non-toxicity. Whether this is with two monoclonal antibodies, two molecules, or four, it doesn't really matter.
I was just trying to quantify, in a way, the possibility of efficacy, of a nanoparticle blocking an extracellular target versus one blocking an intracellular target. I fully agree with your point, and I actually think that indeed the future is in intracellular targeting. But from an economical point of view, if you want, would it be rewarding, going for intracellular targets, considering a possibly lower efficacy?
So let me ask you a question. How many molecules or how much monoclonal antibodies do you think go to the tissue right now with the marketed therapies? How much do you think it goes?
I think it's a minute amount.
Is very, very low amount. So as far as it is enough and the therapy is not toxic, even with the cytotoxic drugs, in general, no more than 2% goes to the tumor, and the rest is around the body. So actually, that is the problem, that it goes over the whole body and creates this toxic effect. So what we could see is that in our experiments is that using a relatively low dose, 4 milligrams per kilo, is a low dose of monoclonal antibodies. Actually, we are now giving 10 and 20 because we think that from the safety point of view, it will be acceptable. And we believe that we can reach much better data. So again, for a particle going into the cell and transporting the monoclonal antibody into the cell is not that difficult. The most difficult part, as it is in gene delivery, RNA delivery, is that once the monoclonal antibody is inside the cell, it has to disassemble from the particle and it has to interact with the target. There is the main challenge now. So still a lot of work in front of us, but I think we have proved a new concept, and this is the use of nanotechnology for delivering monoclonal antibodies inside the cells. More questions, please.
Just one for myself, when I used to do science. Okay, that's a great new concept. Do you see it as a new way to increase potentially the specificity of what we do, the specificity of targeting the cancer cells? Or do you see it more like opening a new, let's say, gateways for targeting new hallmarks of cancer, like classical mechanism that we know cancer cells are addicts and we are barely druggable right now, maybe could be druggable?
Yes, yes, this is the second part of your, exactly. We think that if we can make progress, because this is still in an early phase, if we can make progress in that way, it's going to open a new area in which, you know, nanotechnology can help reaching these undruggable targets. So it's like making new drugs almost, because the monoclonals are there, but nobody's using them because they cannot cross the cell membrane.
Maybe the chromatin dynamics, for instance.
Yes.
Any questions?
Very good point.
Okay, if not, thank you once again very
Thank
much,
you.
Professor Alonso. That was great.